Silvia Capuani is speaking. Please look at the image representative of project development. IMAGO main goal is to develop in vivo MRI with a resolution down to the microscale by exploiting transient anomalous diffusion in MRI, validated and integrated with optical microscopy. As a proof of concept, transient anomalous diffusion MRI on fixed spinal cord was obtained and reported in the final report of project phase 1. The extracted information, validated with light microscopy histology, shows the superior sensitivity to detect submicrostructures of anomalous diffusion maps compared to conventional diffusion MRI. Progress has been made since October 2020 with new results. Single particle tracking confirmed anomalous diffusion of liposomes in hydrogel multiscale porous phantoms. Importantly, we went beyond the phase 1 proof of concept by carrying out an anomalous diffusion tensor experiments on both in vivo and ex vivo mouse brain. Results showed that involuntary motions, such as cardiac respiratory motions, of mice during acquisitions produce image artifacts with a loss of image resolution, preventing from fully exploiting the benefits of anomalous diffusion MRI. We found that currently post-processing motion correction can only partially mitigate this problem. Therefore, a new real-time motion correction strategy based on deep learning is proposed by Sapienza and CNR researchers as the first objective or one of IMAGO Attract Phase 2. This objective is necessary to scale up the IMAGO technology to human investigation on clinical MRI scanners and for taking MRI to a new dimension. We are confident that the results of this first objective, O1, has a great possibility of marketing because an effectual motion correction is a necessary condition to be able to fully exploit any technology that aims to increase the sensitivity, the resolution, and the field of applicability of MRI. The second objective, O2, is the integration of anomalous diffusion MRI and light microscopy on in vivo mouse brain to enhance the sensitivity of preclinical MRI and confirm the performance of IMAGO technology. Toward this goal, 19-fluorine MRI with 19-fluorine labeled fluorophores will be used. Finally, we scale up the technology from animal models to human investigation on clinical MRI scanners. Objective O3. The prototype will concern the IMAGO protocol for the diagnosis of multiple sclerosis, and IMAGO's ability to make an early diagnosis, thanks to subcellular resolution, will be demonstrated. Thank you.
In the first half of SMILE project, carried out in collaboration between CNR Nanotech and ST Microelectronics, our efforts have been directed toward the optimization of molecularly imprinted polymers for the detection of inflammatory biomarkers of oral cancer from saliva and integrated on a surface acoustic wave transducer. The scientific activity during the second part of the project has been focused instead toward the optimization of the microfluidic module for sample handling. This is crucial for sample preparation, above all to meet our vision of a self-contained tool able to perform the sample-in, answer-out workflow, and also of great importance in the prospective management of SARS-CoV-2 contaminated specimen. To develop these aspects, we have optimized micromilled PMMA plastic substrates and the bonding method, holding features of low cost, high transparency, and suitability for fast industrial exploitation. Thanks to the implementation of a micropumping setup acquired with SMILE funds, this work has given good results, which led to the realization of a plug-and-play device for the capture and detection of circulating tumor cells that has been published on Engineering last December. With the aim to improve our commitment to low-cost but high-performant microfluidics, we have also developed a three-dimensional device based on a multi-layered setup with vari-channel and through-holes. As we have described in the newly published paper, this configuration allows the contemporary and real-time monitoring of what happens separately in the two channels and after the mixing point. The entire device can be used as a mixing tool or a gradient generator. At the end of October 2020, we organized EdgeTech Workshop, Emerging and Disruptive Next Generation Technologies for Point of Care, in the frame of SMILE Attract project. We invited high-level speakers coming from academic and industrial context, and we had the opportunity to introduce the Attract program and our SMILE project to a very wide audience. We received several feedbacks among university, companies, and students from all around Europe and more. No less importantly, the workshop expanded our scientific and industrial network, which will support our potential phase 2, adding value for science, industry, and society. A special issue on micromachine with workshop proceeding is also open for submission. So, we are ready to play for phase 2.
Hello everyone, I am here to present the project Multiparametric Magnetic Resonance Approaches for Non-Invasive Glioblastoma Therapy Response Follow-up, MAGRes. We will update you with progress made since last October when projects have finished. First of all, the main results related to our project were accepted for publication in scientific reports in last November. Moreover, we have advanced in the biological validation of the developed biomarker, especially regarding immune system cells profiling, using techniques such as RT-PCR flow cytometry, among others. Under my own supervision, I was the coordinator of MAGRes project. Our colleagues from Universidad Politécnica de Madrid have applied our currently available dataset in order to check whether including information from peritumoral zones, which mean the borders, in addition to tumor itself, using radiomics approaches, would have an added value in response assessment. We have also organized our dataset in order to have it evaluated under non-supervised approaches, which are different from the ones applied in MAGRes, which were essentially supervised or semi-supervised. This will be directed by Dr. Alfredo Velido from Universitat Politécnica de Catalunya, and it's currently in progress. And finally, we have worked towards adapting the current biomarker strategies, which were developed for preclinical formats, but we have to adapt it to human magnetic resonance data formats if we want to include these in a future software with clinical translation potential. This is currently being done in collaboration with ITN InspireMed. If our project was to be selected for Attract Phase 2, so we did not answer again the same questions because they were already answered, most of them, in the last presentation, but we have elaborated a bit more on some of them. So, for example, question 2, with who you would need to partner for this to happen? So, in addition to already mentioned partners in October report, we consider that the main actor that would be needed for scaling up our system would be a medical software company. So we should ensure that all certification, all steps are included in medical device and regulation are being properly accomplished, and we are not missing anything. What applications will you demonstrate with value for science, industry, and society? So we are confident that this biomarker or predictor for effective response in brain tumors will save time for patients and doctors. This is a time that's being currently wasted in the presently used pipeline because they need more or less 3 months follow-up in order to make a clinical decision, a robust decision on whether a treatment is being effective or not. And this wastes time and resources for patients, for the health system, and our system can provide this information in 2 or 3 weeks, even before tumor volumes start changing. So this will save a lot of time. More importantly, no previous knowledge about machine learning or spectroscopic are needed because this will be all integrated in one single software. Regarding question 5, will you count on or apply for other complementary funding sources? And yes, in addition to the Catalan network of health technology, the ShareTech Salute, which was already mentioned in the last presentation, we are preparing to apply for requesting cyber intramural funding to increase TRL level. And also we will apply for Caixa Impulse Validate calls. So being so, thank you very much for your attention.
First of all, thank you for this occasion. I am Maurizio Casalino, coordinator of the Reveal project. The aim of the Reveal project is to design, fabricate, and characterize high-performance silicon-based photodetectors operating at 1550 nanometers, taking advantage of the integration of graphene into silicon technology. Here we have summarized the main advancements since last October. Point 1 and 2. From a technological point of view, we have improved both the quality of the graphene silicon interface and the carrier collection efficiency by realizing a collection metal electrode on the graphene layer. Please give a look at the red dashed line in the inset of the figure. Point 3. As shown in this figure, these solutions led to a responsivity at 1520 nanometers, doubled with respect to the results previously reported, for instance, in the final article required by Attract. Point 4. During the manufacturing activity, we have faced many technological challenges, and thanks to the experience gained during Attract Phase 1, we have conceived and numerically simulated a new structure able to overcome all critical technological steps. In order to scale up our technology, some issues should be addressed. Point A. This issue is strongly connected to a new structure we have conceived. This structure starts from a specific silicon substrate that needs to be developed. Point B. The graphene-based active material needs to be developed for an efficient optical-electrical conversion. Point C. Optical lens for near-infrared optical coupling should be integrated on our photodetectors. Point D. A suitable electronic circuitry should be designed and fabricated. Finally, point E. An efficient chip packaging should be realized. Of course, in order to include these new competencies, the consortium should be enlarged. Concerning point A, we are already in touch with a big company that has confirmed its availability to manufacture substrates in agreement with our specifications. On the other hand, point B is very compatible with the bandpass project developed during Attract Phase 1, and we are in touch with the coordinator, Dr. Monica Beca. Concerning point C, it is compatible with the OptoGlass 3D project developed during Attract Phase 1. Indeed, we required a face-to-face meeting with the coordinator during the Attract Final Congress held in September. Finally, points D and E could be accomplished by research infrastructure communities in Europe. For instance, the Nanoscience Foundries and Fine Analysis RI could be included in the partnership. As already mentioned in our previous presentation, Reveal will demonstrate its value in data communication and near-infrared imaging applications. We add here that Reveal will be able to meet automotive industry forecasts for speeds up to 25 gigabit per second in the next generation of in-vehicle network. In order to contact investor, we will enlarge our consortium with a specific consultant with experience in market management for involving angel and venture capitalists. In addition, early-stage researchers and master school level students will be involved. Concerning the last point, yes, both European and national complementary funding will be considered. Also, crowdfunding platforms will be taken into account.
Our SALT project is a collaborative team effort to develop achromatic optical devices operating in the visible and near-infrared that can be miniaturized down into very small geometries. The basis for the SALT initiative is the discovery by the partners in 2018 of giant broadband refraction in specifically engineered transparent perovskites, KTN lithium, that is, lithium-enriched potassium tantalate niobate. The team includes a nonlinear optics group at the University of Rome La Sapienza in Italy, a crystal growth and development group at the Hebrew University of Jerusalem in Israel, a microelectrode group at the University of Rome Tor Vergata in Italy, and a concept building group at the Simpulse Foundation in Holland. In recent experiments, we have characterized the crystals manifesting giant refraction in collaboration with the group of Professor Stefano Lupi at the University of Rome La Sapienza. Through a spectroscopic study, we have been able to characterize samples as they manifest giant refraction from the terahertz band to the ultraviolet band. Results confirm the absence of absorption resonances and furthermore confirm a broadband giant refraction regime that is even stronger than in the visible from 700 to 1100 nanometers, that is in the near infrared. Using second harmonic generation measurements based on Cherenkov radiation, in collaboration with the group of Professor Luca Tartar at the University of Pavia, we've been able to analyze constraint-free second harmonic generation and corroborate spectroscopic results, showing once again an increased enhanced nonlinearity from 700 to 1100 nanometers. Second harmonic generation experiments have further allowed the demonstration of the switching on and off of giant refraction and hence of enhanced nonlinear response using an external control electric field. Now, the effects of microelectrodes on giant refraction have been studied also using piezo force microscopy. This in collaboration with the group of Professor Carlotta Canalias at the KTH in Sweden. So the basic progress in characterization has allowed us to develop a more profound understanding of the origins of giant refraction. This, of course, allows us to develop more practical ideas on design and implementation of devices. Specifically, we've been able to associate giant refraction to the emergence of a high-dimensional topological phase, whose physical implications are still under study.
Welcome to the update report of our project entitled Combined Optical Imaging and Ultrasound Focusing for Handheld Non-Invasive Cleaning of Implanted Cerebrospinal Fluid Shunting Devices in Patients of Hydrocephalus, Initial Design and Proof of Concept. The aims of our project were to conduct a proof of concept of the feasibility of using focused ultrasound for non-invasive preventive cleaning of obstructions and for the removal of debris in implanted shunts under optical and thermal image monitoring, and to design an early prototype. In the link below, you can see a video with an experimental demo of the successfully conducted proof of concept and of the removal of debris achieved. Progress update in our project can be summarized in 3 main points. Point 1. Advances in prototype design and operational features, including ergonomics, portability, optical components for image monitoring, and other. Point 2 is computer simulations of enhanced setups with several transducers and configurations. And point 3 refers to further experimental tests on standard shunt elements, catheters, valves, and models of patients of hydrocephalus. If our project were selected for a tracked phase 2, we need to extend testing, optimize the design and engineering, and carry out the certification process and the commercialization strategy. We need partners, which include industrial partners, clinical partners, and funding partners. The role of industrial partners is very important since we need a team with experience in ultrasonics technology, computer simulation and user interfaces design, neurosurgical technology, and the regulatory processes for medical devices. Our technology has been designed for a very relevant disease in neurosurgery with a very high social and economic impact. It also shows a strong potential of application in other clinical areas in which catheters are implanted. They may range from oncology to intensive care and other clinical applications. Our technology is clearly aligned with the promotion of preventive therapies, the reduction of impact of acute episodes in the patient, and improving the health system. We have started to promote contact with potential investors through our institutional resources. We also plan to present the proof of concept and the prototype in international meetings related to neurosurgery and medical devices. We have already obtained initial funding from public national R&D calls for the commercialization process of the current system and to explore the possibilities of extension into other clinical areas of application.